Disc 3.
Beadle and Tatum irradiated some 5,000 specimens, then set about testing each one to see whether it could survive on minimal medium. The first survived fine. So did the second and the third. It was not until they tested strain number 299 that they found one that could no longer exist on minimal medium, though, as predicted, it could survive on the complete version. Number 299 would be but the first of many mutant strains that they would analyze. The next step was to see what exact capacity the mutants had lost. Maybe 299 could not synthesize essential amino acids. Beadle and Tatum tried adding amino acids to the minimal medium, but still 299 failed to grow. What about vitamins? They added a slew of them to the minimal medium, and this time 299 thrived. Now it was time to narrow the field, adding each vitamin individually, and then gauging the growth response of 299. Niacin didn't work, nor riboflavin. But when they added vitamin B6, 299 was able to survive on minimal medium. 299's X-ray-induced mutation had somehow disrupted the synthetic pathway involved in the production of B6. But how? Knowing that biochemical syntheses of this kind are governed by protein enzymes that promote the individual incremental chemical reactions along the pathway, Beadle and Tatum suggested that each mutation they discovered had knocked out a particular enzyme. And since mutations occur in genes, genes must produce enzymes. When it appeared in 1941, their study inspired a slogan that summarized what had become the understanding of how genes work. One gene, one enzyme. But since all enzymes were then thought to be proteins, the question soon arose whether genes also encoded the many cellular proteins that were not enzymes. The first suggestion that genes might provide the information for all proteins came from Linus Pauling's lab at Caltech. He and his student, Harvey Aitano, studied hemoglobin, the protein in red blood cells that transports oxygen from the lung to metabolically active tissues like muscle, where it is needed. In particular, they focused on the hemoglobin of people with sickle cell disease, also known as sickle cell anemia, a genetic disorder common in Africans, and therefore among African Americans as well. The red blood cells of sickle cell victims tend to become deformed, assuming a distinctive sickle shape under the microscope, and the resulting blockages in capillaries can be horribly painful, even lethal. Later research would uncover an evolutionary rationale for the disease's prevalence among Africans, because part of the malaria parasite's life cycle is spent in red blood cells. People with sickle cell hemoglobin suffer less severely from malaria. Human evolution seems to have struck a Faustian bargain on behalf of some inhabitants of tropical regions. The sickle cell affliction confers some protection against the ravages of malaria. Aitano and Pauling compared the hemoglobin proteins of sickle cell patients with those of non-sickle cell individuals and found that the two molecules differed in their electrical charge. Around that time, the late 40s, geneticists determined that sickle cell disease is transmitted as a classical Mendelian recessive character. Sickle cell disease, they therefore inferred, must be caused by a mutation in the hemoglobin gene, a mutation that affects the chemical composition of the resultant hemoglobin protein. And so it was that Pauling was able to refine Gorod's notion of inborn errors of metabolism by recognizing some to be what he called molecular diseases. Sickle cell was just that, a molecular disease. In 1956, the sickle cell hemoglobin story was taken a step further by Vernon Ingram, working in the Cavendish Laboratory, where Francis Crick and I had found the double helix. Using recently developed methods of identifying the specific amino acids in the chain that makes up a protein, Ingram was able to specify precisely the molecular difference that Aitano and Pauling had noted as affecting the overall charge of the molecule. It amounted to a single amino acid. Ingram determined that glutamic acid, found at position 6 in the normal protein chain, is replaced in sickle cell hemoglobin by valine. Here, conclusively, was evidence that genetic mutations, differences in the sequence of A's, T's, G's, and C's in the DNA code of a gene, could be mapped directly to differences in the amino acid sequences of proteins. Proteins are life's active molecules. They form the enzymes that catalyze biochemical reactions, and they also provide the body's major structural components, like keratin, of which skin, hair, and nails are composed. And so the way DNA exerts its controlling magic over cells, over development, over life as a whole, is through proteins. But how is the information encoded in DNA, a molecular string of nucleotides, A's, T's, G's, and C's, converted into a protein, a string of amino acids? Shortly after Francis Crick and I published our account of the double helix, we began to hear from the well-known Russian-born theoretical physicist George Gamow. His letters, invariably handwritten and embellished with cartoons and other squiggles, some quite relevant, others less so, were always signed simply, quote, Geo, pronounced Joe, so we would later discover. He'd become interested in DNA, and, even before Ingram had conclusively demonstrated the connection between the DNA base sequence and the amino acid sequence of proteins, in the relationship between DNA and protein. Sensing that biology was at last becoming an exact science, Gamow foresaw a time when every organism could be described genetically by a very long number represented exclusively by the numerals 1, 2, 3, and 4, each one standing for one of the bases, A, T, G, and C. Well, at first we took him for a buffoon. We ignored his first letter. A few months later, however, when Crick met him in New York City, the magnitude of his gifts became clear, and we promptly welcomed him aboard the DNA bandwagon as one of its earliest recruits. Gamow had come to the United States in 1934 to escape the engulfing tyranny of Stalin's Soviet Union. In a 1948 paper, he explained the abundance of different chemical elements present throughout the universe in relation to thermonuclear processes that had taken place in the early phases of the Big Bang. The research, having been carried out by Gamow and his graduate student Ralph Alpher, would have been published with the byline of Alpher and Gamow, had Gamow not decided to include as well the name of his friend Hans Bethe, an eminently talented physicist to be sure, but one who had contributed nothing to the study. It delighted the inveterate prankster Gamow that the paper appeared attributed to Alpher, Bethe, and Gamow, no less than that its publication date was fortuitously April 1st. To this day, cosmologists still refer to it as the Alpha Beta Gamma paper. By the time I first met Gamow in 1954, he had already devised a formal scheme in which he proposed that overlapping triplets of DNA bases served to specify certain amino acids. Underlying his theory was a belief that there existed on the surface of each base pair a cavity that was complementary in shape to part of the surface of one of the amino acids. I told Gamow I was skeptical. Well, DNA could not be the direct template along which amino acids arranged themselves before being connected into polypeptide chains, as lengths of linked amino acids are called. Well, being a physicist, Gamow had not, I suppose, read the scientific papers refuting the notion that protein synthesis occurs where DNA is located, in the nucleus. In fact, it had been observed that the removal of the nucleus from a cell has no immediate effect on the rate at which proteins are made.